It's already 2 p.m. So we all shall start with the last session of today. That is the ninth session of the 5-day long faculty development program. And in this particular session, we will be having two speakers with us. That will be Dr. Mufti Muhammad and Mr. Marcos Ignacio Fabian. And the theme of the session is brain-computer interface, and it will be a continuation of the two lectures that we had on the first day of our FDP. So if it is okay with the speakers, shall we commence with the session, sir? And I'll start with, yeah. All right. So I'll start with introducing the speakers to the audience. I'll start with Dr. Mufti Muhammad sir's introduction. So Dr. Muhammad is an associate professor in the Computer Science and Engineering Department at Nottingham Trent University, UK. Dr. Muhammad leads the MSc in Data Analytics for Business and the Data Analytics strand of the online MBA course. Dr. Muhammad served as Senior Postdoctoral Research Fellow at the Neurochip Lab of the Department of Biomedical Sciences, University of Padova, Italy, between 2013 and 2015. He has been Marie Curie Research Fellow at the Theoretical Neurobiology and Neuroengineering Lab of Department of Biomedical Science, University of Antwerp, Belgium, between 2013 and 2015. Dr. Muhammad's research expertise and interest revolves around advanced machine learning for biological data analysis, advanced machine learning for crowd analysis, distributed biosignal analysis framework, high-resolution brain-chip interfacing, neuroinformatics tools for extracellular neural signal processing and analysis. He is also Grant Review Panel member of Engineering and Physical Science Research Council UK and Ministry of Education, University and Research Italy. He is also member of Higher Education Academy UK, IEEE and ACE. He also has a place in the editorial service, that is a regional editor of Europe's Brain Informatics Springer Nature Journal, Associate Editor of IEEE Access and Frontiers in Neuroscience, Frontiers Journal. Now, I would like to take up the opportunity to introduce our second speaker of the session, Mr. Marcos Ignacio Fabian. He is PhD candidate at Nottingham Trent University under the supervision of Dr. Mufti Muhammad and Professor Ahmed Lotfi. His main research area, machine learning applications in invasively recorded neuronal data for abnormality detection, artifacts, disease staging, and stress detection. Prior to pursuing PhD, he has been a machine learning intern at Spinlock SRL from March till September in 2019. So we are eagerly waiting to hear from you, sir, about more about brain-computer interface. So over to you, sir.
Thank you very much for the introduction and the possibility to present. I'll now share the presentation on screen. Let's see, share sound, share. Okay, we'll start now the presentation. The topic of the presentation in particular is artifact detection and removal in invasively recorded neural signals. I'm an engineer, Marcos Favietti, from Nottingham Trent University. I thought I'd start by showing a few pictures of the university, and Nottingham Trent University has 3 different campuses within the UK, and you're very much welcome to come visit. Okay. To start, let's see the contents of today's talk. We're going with an introduction, mainly talking about the brain, the different neural signal acquisition methods, what are local field potentials, artifacts, artifact removal methods, what is machine learning, deep learning, and the proposed approach. Then the materials and methods, the private and public datasets we've been using for our analysis, the labeling done to the data, and the different neural network architectures we've employed. Third, the artifact detection techniques we have employed, including the different networks and the comparison of their performance. Fourth, artifact removal, including forecasting, a generative model, and the comparison of them. Fifth, a toolbox which incorporates all these elements. We'll go through the different modules and video showing how it operates. Sixth, parallel research, which is some side research that has popped up while we've done the main topic, including multimodality, interpretability, and benchmarking methods, and lastly, the conclusion. Let's start with the introduction. The brain is the most important organ of the body. It's mainly composed of lobes, these being occipital, temporal, parietal, frontal, and the cerebellum. The structural unit of the brain is the neuron, which is composed by the cellular body, and the axon, which fires action potentials. Some of this has already been covered by Dr. Mahmoud 2 days ago, so I won't go too deep into it. However, it's important to know that from these neurons, the synchronity of their firing rate leads to a sum of action potentials, which generate an electrical activity, which can be recorded. These are called neural signals and give insight into the brain's function. Within the UK, there are 14.7 million of neurological cases in England, which include over 150,000 neurological cases. These neural signals come into play because they allow for the study, diagnosis, and follow-up of these diseases. These include Alzheimer's disease, epilepsy, Parkinson's disease, meningitis, migraine, Alzheimer's, and ADHD. We'll now proceed to look at the different neural signals. In this slide, we can see that there are different neural signals based on their temporal spatial resolution and the depth in the image on the right, which basically means the invasiveness. The more invasive the recording is, the better the spatial resolution is. However, the counterside to that is that invasive techniques may potentially harm or scar the user. Therefore, most of the times, signal acquisition is done with the non-invasive ones. The non-invasive ones include magnetoencephalography, which is recorded through capturing the magnetic fields, electroencephalography, which we've discussed the previous days, functional MRI. These three are the ones that compose the non-invasive. And invasively, we have electrocorticography, which is recorded at the scalp level. And we have local field potentials and spikes, which are the most invasive ones. We see from the image on the right that they have different amplitudes. Electrocorticography goes from 0.01 to 5 millivolts and 0 to 500 hertz. Local field potentials from less than 1 millivolt and less than 200 hertz. And spikes from 500 microvolts to 0.1 to 7 kHz. So among all these signals, the ones we're interested in studying are local field potentials. We'll now proceed to describe them. Local field potentials are acquired by low-pass filtering the neural activity of a group of neurons. That is, we capture the overall sum of activities of the group of neurons firing. We can see there are two different signals acquired from two different data sets firing, and the amplitude is around 25 microvolts or 10 microvolts. Like the rest of the neural signals, one thing they have in common is artifacts, which means that an external source of distortion of the signal that generates a disturbance which needs to be removed. In the image on screen, we can see how abnormal waveform looks when an artifact is present. In this case, it's movement. It goes from 10 microvolts up to 1 millivolt, so that's 2 orders of magnitude, or 800 microvolts in the other data set. Again, that's 2 orders of magnitude higher. So artifacts are detrimental to the analysis of the neural activity. They distort it. So they must be identified and removed. This is true for every analysis and application, which includes brain-computer interface. So we'll look at the different artifact detection and removal methods. So first, let's start by looking at the different sources of artifacts. There are many sources of them that can be mainly classified as physiological, which means generated by the subject, or environmental, or methodological, such as the first group, physiological. We have the kind of ocular, such as eye movement and eye blink. We have the cardiac, which is the electrical activity produced by the heart, skin potential, or respiration sources. All of these introduce noise to the signal. On the other hand, in regards to artificial or external, we have light stimulation, the transmission lines, the movement of the electrodes or the subject, and all of these have different bandwidths in which they're present. We can see on the table below. The ocular artifacts are from 3 to 10 hertz. Like the heart one is it's centered around 1.2 hertz. Electromuscle activities from 0 to 200 hertz, which is a problem because that's kind of the range local field potential is, and the other neural signals. So it's kind of hard to remove them. Respiration 5 to 10 hertz, and then we have interference 60 to 50 or 60 hertz. That's a power line frequency or very high frequency. And instrumental, which is the electrode movement, is very low frequency. It's like a slow drift and oscillation. Here we have a video of how a person moving their eyes generates different disturbances, in this case in an EEG. There we have him moving his eyebrows. Again, that's muscle artifacts, and clenching his jaw. We see how it disturbs electrophysiological recordings. Right. So there are different type of artifact removal methods, which start by discarding a corrupted segment. However, it's the worst case scenario because discarding the information, which is time consuming and expensive to obtain, is not what researchers want. Filtering, which can be applied when you have a specific bandwidth. Otherwise, you risk the chance of removing neural information. Template removal is that when artifacts have a unique shape, they come from just one source. They can be approximated and then removed. However, in practice, artifacts come in all sorts of shapes, and the source isn't always clear. Regression and active filtering comes from using a reference channel and then removing the estimated artifact from the signal. The complexity of this is that you need to have one channel per artifact you're trying to adapt. So one for the ECG, one for the muscle artifacts, one for the eye artifacts. So the setup becomes more complicated and expensive. The compositions, such as wavelet or empirical mode, is that they can't remove artifacts if the spectral activity overlaps, which can happen, for example, with muscle artifacts, which are present in a wide spectrum. And so in these all methods have some sort of limitation. So lately there has been a development from advanced computational methods to solve this issue. This is where machine learning comes in. Machine learning are algorithms that can improve their performances using data. Typically, the algorithm has a large number of parameters that can be learned from the data and can be applied in very challenging situations where rules aren't easily defined by hand, such as face detection, speech recognition, stock prediction, and in our cases, the identification and removal of artifacts. Basically, machine learning is a mapping function. That is, the independent variable is converted to the dependent variable. There are many classification models, and one usually associates machine learning and artificial intelligence with complex robot or complex algorithms. But however, linear regression, which is a simple method, which most people in engineering statisticians know, is a machine learning method, because once you have a set of data and you have adjusted a curve, the next time you receive a value, you can predict a value next, you can predict the value in the origin y. So we're mapping, this is a mapping function. So a new data, which we originally didn't know its value, now we can predict with some sort of error the corresponding value on the independent axis. So there are mainly three types of machine learning models. We have supervised learning, unsupervised learning, and reinforcement learning. In supervised learning, we're trying to estimate parameters or class, such as, for example, artifact versus non-artifact. For this, we need to use labeled data and use it to predict the outcome. This is the main one used to identify artifacts. Then we have unsupervised learning, where we have in our data, we're trying to find hidden structures. For example, number of groups. But however, we don't have a direct feedback because we don't know their underlying truth. So for example, it can be used if we know we identify artifacts, but we aren't sure how many types are there. Unsupervised learning can be used. And lastly, reinforcement learning. It's a decision process where an agent is within an environment and learns a series of actions through a system reward. And this it's not mainly been applied to artifact removal. And we can see on the image on the right how Google is teaching an agent to run without giving the instructions, just go from A to point B. And through 0 iterations, it learns to run. Now, machine learning techniques have the requirement that feature extractions are done from the input, in our case, a signal, in order to predict. They're not so good at handling raw signals. And deep learning models have the inbuilt capacity to extract features from themselves and learn. So they're very useful. They're inspired by the neurons. We see the similarity between this is a node in a network with the inputs, the activation function, and the output, and how similar it is to dendrites with the axon and the cell body. There has been parallel development of neural networks through the notions of biology. We have convolutional neural networks are inspired by the receptive fields in cats. So there's a lot of development done in the computational field based on the biology. So on a typical pipeline analysis, the signal was acquired and filtered, and then someone had to manually review their recordings and discard the segments which presented artifacts. Then analysis was made, and then that was turned into a diagnosis or application. What we're proposing instead is that the manual review is done by machine learning artifact detection and removal technique. Thus, the physician or researcher, instead of wasting time doing the manual review, can spend that time doing the analysis and the diagnosis and application, improving results. So let's go to the materials and methods now. We used 2 data sets. One was private and one was public. We have in the first one on the left, it was recording on both our local field potentials recorded in rodents. In the first one was freely moving rodents, and in the public data set, what was rodents in a treadmill. where we have the physicians have labeled epochs, so times where no artifact was present in the signal in both of them. And we have used, let's start again, how we label the signal. Well, we have here two examples of windows with their corresponding power. So that's the activity, the sum of the activity to the power. That's how the power is calculated. So it's millivolts square. And we slide a window of X duration. We see that there is a threshold that the normal activity doesn't pass. So if we apply the same threshold for the whole signal, everything that goes above it can be considered as artifact, and everything below is considered null. And that's how we proceeded to label the data. In regards to neural network architectures, we use a multi-layer perceptron, which is one of the most simplistic neural network types with sigmoid dimension functions, a recurrent neural network, which takes in consideration previous time steps. In particular, we use a long short-term memory network because they solve the issue of vanishing gradient. And lastly, we use a convolutional neural network, which is mainly composed by the following layers: a convolution, pooling, dropout, and fully connected. And these can extract temporal characteristics and local characteristics. So they're very good for analyzing images. However, we use a one-dimension variation. So we'll start first by our research done in the artifact detection. The first research we did with multi-layer perceptron was that we had 5 rodents, and we wanted to see how well a model trained with one rodent could predict the others rodents. So we see here on screen these matrices, which represent how well a model trained with the rodent name on the column on the left predict the rodent on that name is on the bottom. And A, B, C, and D correspond to different window sizes. So this is 50 milliseconds, 100 milliseconds, C is 150 milliseconds, and D is 200 milliseconds. So we see that the best result is obtained by 150 millisecond results, where a model trained with individual rodents can predict very accurately the normal or artifactual activity in the other rodents. In addition, we created a global model, which means we mixed the data from all of the rodents, and we've had an average accuracy of 93.2%, and the computational time for a window size of 150 hertz of 1 minute of classification is around 2 seconds. So that's the best compromise between accuracy and computational time. The next analysis we did was classification using long short-term memory. Long short-term memory are a sort of recurrent neural network, which is a type of network that have a hidden state. The problem with the hidden state, which captures previous states of the network, is that the vanishing gradient problem, where applying it many times makes the gradient go nearly to null, which the LSTM solves by making it the hidden state equal to 1. The results of different LSTM networks shown in the table on the right show overall accuracy of 87%. Therefore, we tried another approach, which was the forecasting error, which basically means if we have a window that slides through the signal, then we're trying to predict the next point, whether it's we're trying to predict normal behavior in a signal. So whenever we go to an artifact section, our ability to predict accurately should be worse. This is measured by the root mean square error. However, we found that the distribution curves from normal points and artifact points overlap significantly. So we couldn't find a major difference in the error between forecasting normal and artifacts. So this approach didn't give any good results. Lastly, we compared different convolutional neural networks where AlexNet inspired network achieved a 95.1% accuracy, which is better than LeNet 5 and BCG16. We see that the different training accuracy and losses, where the AlexNet achieves the best loss. And then we also used the property of convolutional networks with GradCam activations, which means we can visualize class discriminative features. In particular, we see, for example, the non-artifact correctly classified. It looks at many different values. However, for artifactual signals, we see that the network is looking at the very high amplitude peaks. So, and also the incorrectly classified, we can see what the network was paying attention to when making the classification. Now we compared the different data sets with all three techniques, where we find that the 1D CNN in both cases have achieved the best performances, and the computational time is the best. So we have an algorithm which performs accurately and fast. Therefore, we can conclude that the 1D CNN is the best model for this specific task, which is artifact detection. We proceed now to artifact removal, where the pipeline which we're presenting is once we have defined normal and artifactual segments, what we'll try to do is forecast normal behavior. That is, the network will learn to predict how normal behavior is, and then replace artifacts with that normal behavior, and then we'll compare. We'll try to see if we can fool the artifact classifier, and then use the parallelogram to study similarity or difference between them. So forecasting, as I said previously, is taking a window at a time t to predict the time t+1, and then incorporating the predicted value t+1 to predict the value at time t+2. We used a CNN LSTM structure with 16 layers, and for the sequence input, we use from 100 to 900 points, and then for the output, we predicted 1, 5, 10, 25, 50, and 100 points. The results are compiled in this table, where the best performance for the LSTM network was 0.773 root mean square error, and for the CNN LSTM, the best performance was obtained with the 600 data points of input and 100 data points of output. We compared it also to RMAX, which is a statistical method for forecasting, where we see that the root mean square error of the neural networks is significantly lower, and the prediction time is significantly lower as well. However, these two methods do require a lot of computational power and time to train. As a contrast, we also did a generative network, or GAN as they're known, in which where we used a generator network and a discriminator network to try to train a generator to create normal examples. So basically, the generator takes a random input and generates a sample, and a discriminator is trying to discern between if that example is artificially created or comes from our normal LFP population of our data set. And these two models, the generator and the discriminator, are competing against each other. The layers are here on the table below. We see that after several iterations, the networks converge. The result of a model we did trained with artifact and the real dataset normal classified the generated normal activity with 98% accuracy. So that means that a dataset trained with our original dataset classified the new synthetic examples as normal 98% of the times. And then another model trained with the artifacts of our dataset, but with the normal synthesized examples, classified the real normal examples 88.5% of the time, which means that we have achieved overall a good result. We now proceed to the comparison of these methods. The first one is, well, a plot with the parallelogram. So we see how the physiological, the generated, the artifactual, and the replaced signal of 1 second duration are. And then the spectral activity, where we see that the major difference in the artifacts are at the lower frequencies of the parallelogram, where the activity goes to nearly 1 decibel. This is on the log scale. And both the replaced and generated look quite more similar to the physiological activity. If we look at the mean power per band, we notice that the physiological activity on blue, the artifacts on red, the generated on yellow, and the replaced on green. We notice that the generated band is quite more closest to the physiological band in all the different frequencies. And lastly, we have this plot of the TNSC features, which is mapping all the features to a 2-dimensional space, where we see there is in red the artifact, which is the furthest away, this generated ones in yellow, and it's replaced ones in green, which overlap significantly with the blue. That means that the replaced and physiological normal signals have the same characteristics. Right. We then compiled all of these signals into a toolbox. So we basically developed this toolbox to complement the gap in the literature and the available toolbox, because not a lot of them had artifact detection. Some of them had digital filtering. Some of them, every one of them had data visualization. Some of them had spectral analysis. Almost none had artifact removal nor file operations. And most of them are able to handle multiple formats. We also have a plot diagram of the 4 different modules this anti-app has. The first model in yellow is the labeling. The one in green is the training artifact detection network. In blue is forecasting, predicting new data for identification of artifacts. And in purple, we have the artifact removal module. So for labeling, we have, this is the first module, which we can load signals and then format into a structure, into a table, which allows the machine learning models to analyze, which allow introducing the sampling frequency, the window length we wish to analyze, and we can also scale the data. And then once that is done, we can select a threshold per channel. We have two tools for that. One is a table we can sort with all the information and the power of the channel, so we can just sort through there and analyze. The other possibility is by an histogram plot, which then can help us find where the histogram deviates and set the threshold based on that. And we can also plot the different windows. For artifact detection, we can load the training data once labeled, split it into training, validation, and testing, which is commonly done to not overfit the data. And we can also balance the data, because artifacts are usually such a rare occurrence. It is custom to downsample the amount of normal examples to not introduce bias to the data. We can use then choose different networks, the MLP, the 1D-CNN, or the LSTM, or load one of our own. We can create a network and then use the training options, such as the solver, the initial learning rate, validation frequency, the max epochs, the mini batch size, execution environment, if you want to plot, and also some more advanced, which are mirrored from the neural network toolbox in MATLAB. And then we can train the network and then obtain different results, such as the confusion matrix, the AUC score, and the ROC curve. And then we can also set a custom threshold for classification. Lastly, we can save this training model and then load new data to accurately predict whether a segment is artifactual or not. In regards to artifact removal, this is the fourth and last module. We can load the training data, extract normal segments to train the network, which will forecast the normal segments. And then we can extract from that segment how much input we want in the network, how many points we want to predict, and how much data points left over are used for testing. We can also choose between if we want milliseconds or data points. And lastly, we can split into training, validation, and testing, and train the network and view the different results. And we can then use, if we're happy with those results, we can replace the segments in the channel. As we can see here, the artifacts have been reduced from all the way from 0.5 to 0.05, so an order of magnitude. We'll now see a quick video showing the performance of the artifact removal module. So first we load the data. Then we extract the segments and train the network. And now we're looking at the different examples of the original value and the one predicted by the network. And if we're happy with it, we can then proceed to replace the segments. And now we can see that the high amplitude artifacts have been removed. Right. So now I'd like to take some time to talk about the parallel research, that is the other research that has appeared throughout the main one. The first one is the benchmarking of artifact detection and removal. We've compiled over 100 articles which deal with artifacts in electroencephalography, magnetoencephalography, electrocorticography, local field potentials, and neural spikes. The analysis showed that the main signal used is electroencephalography, followed by magnetoencephalography, and then ECOG LFT spikes on similar fashion. This is mainly because the electroencephalography is non-invasive, so it's easier to acquire. In regards to the artifact type, we have that the main one used is the main one studied is the ocular one, due to that it greatly disturbs electroencephalography, followed by muscular and cardiac, and then we have power line noise, random noise, and others. In regards to the machine learning methods, we have the 39% use neural network, 30% use support vector machines, 12% use clustering, and 9% use other. We have also studied how these different artifacts are studied together. So the main combination of artifacts is between electromagnetic muscle and eye artifacts are studied together, and then eye, muscle, and heartbeat studied together. In addition, we also plotted this relationship between the different artifacts and the different methods, where we see that the ocular artifact is mainly studied by neural networks and support vector machines, and the muscle artifacts are instead studied a bit more by support vector machine and neural networks. So with this plot, we can see the relationship between the method and the artifact. In addition, we have developed also a benchmarking artifact detection tool. The tool allows for the user to input the characteristics of their method and the signal they use, and they can filter throughout the whole literature to find out how well their method works. It's composed of 3 slides, 3 tabs. So the first one allowed to filter, and if you hover over the data points, it tells you the characteristics and the author of each paper, and the characteristics which we chose are the number of examples used to train the model, the features extracted, the hyperparameters of the model, and then the normalized performance. In addition, this information is also presented in the form of a table, if that's easier to browse. And lastly, the citation of the author, so it can be easily found in the literature. Now we proceed to multimodality in artifact detection. A way to address the issue of noise is to incorporate another source of information to the system that allows to identify artifacts. The combination of different formats is called multimodality, and is widely used in other neuroscience areas, such as neuroimaging, where multimodality provides clinician with a metric of the relative health of this function of the brain. In the research we did, we looked at the pros and cons of adding a second source to detect artifacts. And we looked at body movement, head movement, eye movement, and blink. I looked at muscle artifacts, and heart artifacts. So the examples of reference, we have the ECG, typical study recording of the electrical activity of the heart with the different leads. This is some electrodes put on the face to detect ocular movement. We also have, this is a gaze detector, which can also detect eye blinks. This is a portable version of the same device. And then we have an accelerometer, which can help us detect sudden movements. In summary, we found out that the advanced computational methods have helped a lot with the replacing these reference channels outside accelerometers, gyroscope, and multimodal sensors, as these are now already incorporated in the technologies. However, if they're available and they don't disturb the user, it's always a great benefit to incorporate because it makes the recording more truthful. As a negative, the incorporation of another channel makes the setup more expensive and can also limit it to a laboratory setting, which is not what we want, for example, for PCI devices. We also explored the open data in invasive neural recordings. While datasets found online range from a few kilobytes to several terabytes, we searched those from intermediate size of 1 to 10 gigabytes. The inclusion criteria was based on a poll made of different scientists and data scientists. And we found that data from 11 repositories, a total of 27 data sets were explored, where we find that the main animal studied from was rodent, and the main signal study was local field potentials, and that the most commonly used format was .mat, so the MATLAB file, which indicates that for neuroscience, it's useful to know MATLAB. In regards to the challenges of data sharing, we found that there's limited descriptions of the process used, integrating different file formats, identifying the protected health information, adhering to government regulations, and a lack of platform are the main challenges. The needs are standardization of the data, basically, especially of the free text notations. The negatives are that researchers fear data sharing may lead to question the validity of the analysis, so researchers may be hesitant to share their data. And moving forward, there's new platforms such as GitHub and Kaggle, which are allowing to share more data, and aggregating small data sets can help get new insights. In regards to the data sharing format, if we remember our survey, we found out that .mat was the preferred sharing type, and this matches a survey done by the Berkeley Institute of Data Science, which conducted a research with 440 responses from 90 unique universities and institutes, where the most commonly used format was .mat. And lastly, some difficulties we find is that locating the data set it's kind of difficult without using the correct keywords or prior knowledge of available repositories. There are some quality problems such as very small data sets, duplicate or wrong values, the format of the data not being easy to manipulate, having to match and standardize that data when using multiple sources, and structure. And in addition, the metadata and description of the experiments, it's mostly behind paywalls, which means that even though the access to the data is free, to be able to understand the data, it's not. So that may restrict the understanding of it. Another analysis we did was the impact of the sampling rate in invasive neural recordings classification. So we looked at, in this case, electrocorticography. We looked at the available open access data sets, and from them we chose the one with the highest frequency sampling rate in order to compare, have more range to compare. In this data set acquired from Kaggle, the data was filtered in a bandwidth of 2K hertz and downsampled to 5K hertz. The recordings were classified into these 4 classes: physiological, pathological, line noise, and non-cerebral artifacts, and then segmented into 3-second sequences. So here we're kind of combining, studying the detection of artifacts with how the sampling rate impacts it. So basically, we have this data set is composed of 2 sub data sets. We have balanced the classes because they have different examples per class, and we don't want any bias to any one of them. We then resample them to different frequencies to 250, 500, 1K, 2K, and 5K hertz. Then we apply the normalization to the examples and then do our research with the 1D CNN that had provided good results before. In the image, we see the difference between 5K hertz and 250 hertz. So clearly, while the shape is still there, some high resolution details have been lost. Now on screen, we have the adaptation of the 1D CNN version of AlexNet, which was achieved by flattening the convolutional layers. The main layers are convolutional, pooling, dropout, and fully connected, and it has 12 layers. On the table, in brackets, is the filter size multiplied by the amount of filters and followed by the stride. We show now the different training plots where we see that at the beginning, the 5K hertz converges quicker to the result with very few iterations. Meanwhile, the others have a slower learning curve. And however, after several iterations, all models converge to a similar result. In regards to the test set, what we find for each one of the frequencies is that an overall difference between the highest frequency rate and the lowest frequency rate of about 2.7%, which isn't a lot compared to the gain in computational speed and storage. And the detection of artifacts, which is class 0 and 1, is maintained consistently high. If we see they're 99% and 91%, and in the lowest frequency is again 99% and 87%. And if we compare these results to a similar architecture applied to analysis of echo signal, a model like that consumed 802 microwatts per second per detection, which means that this model can be ported to a device, and the CNN in itself can be reduced 7 to 8 times by reducing the bit size of the weights. Overall, the benefits of a reduced sampling rates are less storage consumption, smaller machine learning models, and less energy consumption in the portable system. There are also development in techniques to reduce the size of transmitting the signal, so even more faster can be achieved. And for our model, there is the optimal frequency rate would be 250 hertz due to the high performance and slow dimensionality. When compared to a human, humans detect accuracy in detection doesn't improve above 100 hertz. So there's still a lot of room to improve in that regard. However, humans can compete with machines in terms of speed. Now we move forward to the portability of machine learning models for neural recording. Continuing the last concept of small models to apply in portable devices such as brain-computer interfaces. In this one in particular, we look at electroencephalography, because due to after reviewing the literature, we found that it was the most the signal which studies mostly addressed due to it being non-invasive. The computational methods allow to process the streams of data, such as the machine learning models we discussed, and the portability of these models can be challenging. However, a lot of machine learning models have been applied in portable EEG systems, and we can see the pipeline in the figure on the left, where it starts with acquisition, transmission via Bluetooth or Wi-Fi or cable, the preprocessing, which includes filtering and structuring the data, feature extraction, such as principal component analysis, and then the classification with these portable systems. When looking at the different applications, this has been portable devices have been used, include artifact removal, death of anesthesia, drowsiness, the detection of eye movement, emotion detection, motor imagery tasks, which is mainly for brain-computer interfaces, movement prediction, Parkinson's state, the detection of the different Parkinson stages, pattern recognition, seizure detection, and sleep status. We compiled all the studies in the literature where we find that the most studied application is seizure detection with portable machine learning systems with 18 studies, and artifact removal, for example, which is our interest, has been applied 4 times, and motor imagery task 3 times. We also see that the most used technique are neural networks with 42% of the total, followed by support vector machines with 37%, and then linear discriminant analysis with 7%, and between 2 to 5% linear regression, decision trees. Among the different neural networks, we have CNN and functional neural networks are 44% each, and LSTM and artificial neural networks 6% each. In regards to the power consumed per application, we plotted it in regard to we plotted the power consumed by each method in regard to the application and the method. Basically, it's hard to see a trend among them. We can see, for example, in the power per application, seizure detection is across all the board, and artifact removal is a bit on the higher end. And some of them just have a very few examples to find a good trend. And in regards to the power per method, we do see kind of a trend here where convolutional neural networks are a bit on the higher end. Linear regression is one of the simplest methods, so it's a bit on the lower end. Functional neural networks are a bit on the lower end as well, and LSTM network in the middle. So basically, it's kind of hard to find a relationship between the power application or the method. Nonetheless, this study may help those trying to see something, if there are other studies like the ones they're trying to carry out. So what about the challenges? They are data labeling, which is how to officially tag the data where short events happening in long duration recordings. Explainable AI, such as the development of methods which allow interpretation of the black spot machine learning models. Multimodality, which we have mentioned previously, which is that by adding another source of information, the model performances can be improved. However, these require a much more complex pipeline and consume more resources. Compatibility and scalability. These machine learning models work in synchronism with communication, storage, and display. So sustained compatibility and communication protocols are required. Privacy, how the patient's data will be protected or encoded during each of the steps from signal acquisition all the way to storage. The storage, both from how the data will be stored, as well as where, in the sense of internal memory, local server, or cloud, and the format. User variance, in the sense that we're complex beings which change over time due to lifestyle or metabolic changes, or the presence of a degenerative disorder. And patient-to-patient variance, as people have different brain patterns, user-specific models are not as effective when generalized. Okay, lastly, interpretable models. We research interpretable models via feature extraction and decision trees. Basically, when using a machine learning model, we don't know how the machine learning model is making the prediction. We can see the outputs in the majority of them, but a human can't understand the mapping process. However, with interpretability, there are some models which allow us to gain insight into the decision process and then verify the labels, and with our feedback, we can improve the results. Within neuroscience, the application of machine learning model has been used to classify patterns, control brain-machine interfaces, and gain insight into the brain. And it's very challenging in the healthcare area to use these models where no feedback is done. So it's very important to have interpretable models. So what we proposed is basically from the private data set. We used the same 150 millisecond bins we used at the beginning, and then we extracted different features and compare their performance, and then created a joint feature set, and then ranked the features in their performance to find the best set of features. And what we use as a classifier are decision trees, because coming back to interpretability, decision trees are a method which have an innate property in them, which allows the user to understand the decision process, and it's quite familiar to the rule-based system physicians and doctors use. So first we labeled the data. We have seen this bit. We extracted features from 3 different toolboxes. The first one has 30 features. The second toolbox has 26. And lastly, we use autoregressive features of the 6 orders. So that's 6 features. In order to rank the features, we use 3 different techniques, 3 different measures, which are mutual information, minimal redundancy, maximal relevance, and then Pearson correlation. All of these are available out of either free access toolboxes or are already included in MATLAB. So let's talk about a bit more about decision trees. The trees are split using the Gini index as a splitting criteria, and the obtained tree is pruned by cost-complex pruning. The algorithm can be described in the 4 stages: tree building, stopping the building, pruning, and optimal tree selection. In the first stage, a tree is built using a recursive splitting of nodes based on the distribution of the classes learning the dataset that would exist in that node, as well as the decision cost matrix. Each resulting node is assigned to a predicted class. Whether or not a node is broken into a child node later, a predicted class is assigned to it. The second stage consists of stopping the growth of the tree, as a tree which probably greatly overfits the information contained with it in the learning dataset has been produced. The third stage results in the creation of a sequence simpler of trees, though cutting off increasingly important nodes. In the final stage, the tree which fits the information in the learning dataset but does not overfit it is selected among the sequences of pruned trees. Binary decision trees are easier to interpret than other machine learning models, as the decision of each node is easy to interpret. Well, looking at the results we obtain, we first see the classification results obtained by each one of the toolboxes, where the best score was obtained by the EEG-fed toolbox, despite that it was a toolbox to extract characteristics from EEG and worked on local field potentials. We then proceeded to rank the features. We can see on screen the different rankings where mutual information and Pearson correlation have ranked the top 15 features very similarly, and however, MR have ranked them noticeably different. The performance of the decision trees trained with 1, 5, 10, and 15 features is shown on screen, where MR has the best performance with 1 feature at 88.1%. However, using 5 features with mutual information achieves almost the best score, and then with 10 and 15, it does with 89.3%. So we have that the best method is mutual information for ranking features in this particular task. Comparing with other methods, the ones we have shown previously, for this data set, we found that decision trees work better than the LSTM network, slightly worse than multi-layer perceptron, and well, there's quite a bit gap with 1D-CNN, which is a very powerful technique. However, the training of the 1D-CNNs consumes a lot of energy and power, while decision tree doesn't. And for example, decision trees allows us to follow the classification of one example, as seen on screen, and see the decision process between each and every single one of them. So it's locally and globally integrated. We can see how the whole system makes the decision, and then how the individual example makes the decision. And instead, the 1D-CNN, we need to rely on either silencing maps, CAM or GradCAM to understand where the individual example was classified. As an example, we show the GradCAM of the 1D-CNN, where the normal example has been classified. And we can see in yellow the areas which the network is paying attention to classify it as normal, and blue, dark blue, the ones it isn't looking. So yeah, we have achieved a good model which is interpretable and allows the identification of how every example is classified, with a performance relatively similar to a neural network. Conclusion. We're almost to the end now. We set out to study whether machine learning can be used for artifact detection and removal. We achieved accurate results with different models and obtaining 2 datasets. The research was embedded in a publicly available toolbox for the community, which is the Santia toolbox, which has been published in a journal, and also the artifact removal and benchmarking, which is under process. The generated expertise will be used to detect stress segments in animal model of Alzheimer's, which need to be identified and removed before a study. And overall, this research has generated 10 conference publications and 3 journal publications, 1 published, 1 under review, and 3 ready for submission. Thank you for your time. If you have any questions, please write them on the chat. We'll now proceed to the Q&A session.
Thank you very much, sir, for this wonderful session. I would request the participants, if you have any questions, please post it in the chat box, or you can even unmute yourself and directly ask your question to sir.
No.
All participants have been given the unmute permission. So if anyone wishes to ask your question, please unmute and you can ask. Right, I don't think there are any questions coming up right now. So I would like to take the opportunity and thank you, sir, for giving us a very thorough idea about artifact removal detection, how machine learning is playing a very important role, what are the different machine learning algorithms which we are using throughout this process. So thank you very much. It was, a lot of the information was brand new for us. So thank you very much for sharing this information. And thank you.
Thank you very much for inviting me to give this talk. I'm very honored, and I hope some of the information I've given you you have found useful.
Absolutely. I would take this opportunity and I would request Professor Tufan Shahas to please share the virtual plug. Sir, since this is a virtual meet and we are not able to meet and greet you physically, so we have prepared a virtual plug for you, which will shortly be shared. Yes, it's coming up. Yes, sir. So please accept this small token of appreciation from our end. This is especially for you, and thank you very much for all your time in this particular faculty development program.
Thank you much for the certificate. Looks very nice.
Thank you, sir. So with this, are we coming to the end of the session? In the meantime, if any participant has any question, please unmute yourself and you can directly ask the question to the speaker. All right, I don't think there are any questions coming up. So in that case, with your permission, shall we end this session, sir?
Yes, please.
Okay, thank you, sir. Thank you very much. So to all the participants, please find the attendance cum feedback form in the chat section of this particular Zoom meeting. This Zoom meeting will be on till 3:30 p.m. and all the participants are requested to give your attendance by 3:30 p.m. The form will be open till 3:30. And again, thanks a lot to our speaker for the day. And thank you very much for your input. Thank you.
Yeah, thank you. Goodbye.
Bye.